Today we'll be studying one topic, which is the chemical digestion of all of our foodstuffs. There are four types of foodstuffs that we eat: carbohydrates, proteins, nucleic acids, and lipids. And we're going to be studying the chemical breakdown of all of them. And I've thought a lot about how to teach this lesson through the years, and I guess it comes down to two basic steps. We take something that is quite complex, what we're going to call a polymer, the root poly meaning many, the root mer meaning like things, structures. So polymers are going to be broken down into oligomers and dimers. The root oligo means small number, but more than two, so somewhere like 3 to 8. And dimers, of course, means 2. So oligomers and dimers would be sort of like an interim stage, and then monomers would be a single stage, mono meaning 1. So when we have monomers, then we are actually able to absorb these things. So you may recall that when I discussed chemical digestion in the previous video, I said to keep in mind two different terms, one of them being enzymes. And for enzymes, you're going to be looking for the root ase at the end of the word, or the root in, which indicates a protein, but in this case, all of the ins we're looking at today are going to be enzymes. So enzymes would be things like lactase or pepsin, for example. So ase or in. Okay, so two key words with chemical digestion: enzymes, and then monomers, which is a unit of foodstuffs that's small enough to actually be absorbed. Oligomers and dimers are still too large to be absorbed. It's an interim stage, but monomers are small enough that at this point you can actually absorb them into the body, usually through the walls of the small intestines. All right, let's look at a more concrete example of this to make it a little bit more palatable, literally palatable. We're going to start off with carbohydrates. There are polysaccharides that we primarily eat, which are starch and glycogen. Starch is the way that polysaccharides or complex sugars are stored in potatoes and other vegetables primarily. And then glycogen is the way that animals store complex polysaccharides. Now, these are going to be broken down in the first stage into smaller units called oligosaccharides and disaccharides. So disaccharides, of course, meaning 2 small sugars still attached to each other, and oligosaccharides meaning, between 3 and 8 sugar units still attached to each other. The disaccharides we primarily deal with are maltose, sucrose, and lactose. You see that OSE at the end of the word? That means sugar. Sometimes that's a little bit mixed up with the ASE, so just be careful of that. And we end up with monosaccharides, glucose being the biggie, and then there's also galactose and fructose as well. So these are the 3 monomers of sugar that we're going for, that we're trying to break all of our carbohydrates down into these 3 monomers before they can be absorbed. So let's be specific. When we go back up to our general formula, then we are going to be drawing our first set of enzymes or structures in blue in each case, and we're going to be doing the second set in orange. So the first set of structures would sometimes be enzymes, but there is also a few incidents where it's not an enzyme that does the first digestion. It's going to be something like an acid or a detergent. Well, our first example is pretty straightforward. When we talk about carbohydrates, there's really only one enzyme that does that first digestion, and it's called amylase. And there's 2 places where amylase is secreted in the body. One of them would be in the mouth as part of salivary amylase, part of your saliva. And the other place that we have amylase found is in the pancreas, so pancreatic amylase. So there's salivary amylase and pancreatic amylase. And as far as I know, they're actually pretty much exactly the same structure, molecularly speaking, other than the fact that they do their work in 2 different parts of the body. So your first digestion of complex carbohydrates is going to be in the mouth. It's one of the reasons they say to chew your food really well, because you actually do, if you leave food in your mouth long enough, you actually will do a great deal of your carbohydrate digestion right there in the mouth, and it'll be a little easier down the line for you to continue to digest your food. Pancreatic amylase is going to do its work actually in the small intestines, and we're going to talk about that in a little bit. Right, our second set of enzymes is going to usually be a brush border enzyme. Let me explain what that is. So we're going to draw a villus here. The villus, you might recall, was in the walls of the small intestines, And along the outside wall of the villus are these little structures called microvilli. Well, microvilli actually look a lot like a hairbrush, like bristles on a hairbrush. And so this is called the brush border. So enzymes that are secreted by these microvilli are called the brush border enzymes. And so the examples that we look at here, and they are all brush border enzymes, would be quite specific in some cases. Like for example, maltase is an enzyme that breaks down the dimer maltose. Sucrase breaks down sucrose. Lactase very famously breaks down lactose. The only way you can get rid of your disaccharide lactose is if you have lactase present. So people who are lactose intolerant, oftentimes it is the case that they do not produce enough enough lactase, or maybe just a smaller amount than is needed. Which, by the way, just really off topic here, it's just natural, evolutionarily speaking, it's actually natural not to produce lactase because, you know, in evolutionary time, way back, thousands of years, we actually never ate, we never drank cow's milk after adulthood. We had different types of proteins that were present to break down milk sugars when we were babies, and then after we become adults, we didn't consume dairy anymore. So it's relatively recently that we have adapted the ability to continue to produce lactase into adulthood so that we can continue to eat ice cream, for example. And then the two other enzymes that are a little bit more non-specific would be dextranase and glucoamylase. All five of these enzymes are secreted by the microvilli in the small intestines, and so they are all brush border enzymes. And so in my opinion, the carbohydrate story is a fairly simple one. You basically start off with the starch and glycogens, you have this intermediary structures, and then you have, you know, quite specific types of enzymes that break down the most complex into less complex, and then the less complex structures into our monomers, which we can then absorb. So let's move on to discuss proteins, our next of our foodstuffs. Proteins, I'm going to draw a little protein here. Remember, the proteins look kind of crazy, but in some time ago we discussed the structure of proteins. It's mostly made up of alpha helices and beta sheets that are kind of structured together and kind of folded together in kind of unique ways, and so it looks crazy. But what we want to get at with proteins is we want to get to the point where we are able to pull apart what looks like really tangled threads and just craziness into something that we can actually break down, because man, our bodies just love amino acids. You know, this is how we build our own proteins. So we take the proteins out of animals and plants that we eat, we break down those proteins, and then we are able to digest down and absorb those amino acids to build our own body's proteins. It's quite important for us to have access to all those proteins. So the first thing we want to do is we want to denature the proteins. So when we denature a protein, if we took, for example, an alpha helix and stopped coiling it so closely together, you might end up with a looser coil like this. The best way to denature a protein is with an acid such as hydrochloric acid. And then when we have sort of like this primary shape of the protein instead of this quaternary and tertiary shape, and it's denatured, then we can start to nick away at that primary shape with another protein, another enzyme called pepsin. Now, hydrochloric acid is not actually an enzyme, it's just an acid. It isn't a protein or any kind of a structure. It doesn't, for example, end with an ASE or an IN, but it does work really well to denature proteins. It's one of the things that makes it so harmful to humans, actually, for the most part. But pepsin actually is an enzyme that is secreted by, we know this to be true from the last video. Hydrochloric acid and pepsin are the structures that make up gastric juice, which we secrete by the gastric glands in our stomach at the order of 3 liters per day. So hydrochloric acid and pepsin together will do a pretty good job of breaking apart those proteins. So now we have proteins, and we're going to kind of follow the same shape that we had before, but now we're going to keep in mind that pepsin went ahead and broke this thing apart, and so we have these little pieces of a protein, little chunks of a protein. At this level, you would be looking at an oligomer or a dimer, and you would be trying to break this down into smaller pieces. And for this, you would use, there's a few different enzymes that we use for this, but the main ones are trypsin, sorry about the spelling there, trypsin, and chymotrypsin. These are both secreted by the pancreas. And keep in mind I'm using the blue ink here because this is only the first stage of the digestion. We're taking fairly large structures and still breaking them smaller. So chymotrypsin and trypsin will do that. They both come from the pancreas. And now we're looking at pretty small chunks, and we're going to do our very final digestion of these, where we're going to be breaking in these, fairly small chunks at this point into even smaller pieces. So they would be called oligopeptides when they're in smaller pieces. Remember, oligo meaning a small number, somewhere between 3 and 8. Peptide bonds, and peptide bonds are where you have amino acids attached to each other. So the very last thing that's going to happen is you have these brush border enzymes, aminopeptidase, carboxypeptidase, and dipeptidase. These all are secreted by the walls of the small intestine, those microvilli, and so they are brush border enzymes. And finally, you end up with your monomer, which is the amino acid. Now, all of the monomers of the carbohydrates, glucose, galactose, and fructose, are absorbed into the blood, and amino acids are also absorbed into the blood. In this video, we're not going to be going over the details of exactly how things are absorbed, but I did want to make the point of saying that they are eventually absorbed into the blood. These are fairly large molecules still, so they have to be transported. They oftentimes have co-transporters or active transport allowing that to happen. But at the end of the day, they're absorbed into the blood where they can then be utilized. Carbohydrates, of course, are used by our body. Oftentimes, all carbohydrates are converted into glucose, which is used, of course, in the Krebs cycle inside of the cells in cellular respiration. And then amino acids, again, are used as building blocks for the body's own proteins. Well, let's move into our next type of foodstuffs. So we finished with carbohydrates and proteins. We're going to move on to nucleic acids. And nucleic acids are DNA and RNA. So we'll just draw a quick little strip of DNA here, crisscrossed like this. And at the very end of this, we're going to have I had to kind of squeeze it in the corner there. We're going to have a little pentose sugar with a little oxygen there. Hanging off one end of the pentose sugar is going to be a phosphate group, and then hanging off the other end is going to be that nitrogenous base. And the nitrogenous base, if this was DNA, it would be an ACGT. If it was RNA, it would be an ACG and U. Those would be the options for that nitrogenous base. So we're not going to be drawing out all the details here, but I thought I'd go ahead and draw another one over here upside down, indicating the 3 to 5 versus 5 to 3 configuration of each ladder of the DNA. So these are nucleotides. So I've drawn out 2 different nucleotides here, which are comprised of 3 parts, of course. Technically, the nucleotide is the monomer of the nucleic acid. And the way that we cut apart these nucleotides is with an enzyme called nuclease. Now, the nuclease comes from the pancreas. However, it does its work inside of the small intestine. Let's discuss that, because that's the this is the 3rd time that we've actually come across something that we said, okay, it came from the pancreas, but it does its work in the small intestine. So, for example, if we looked at pancreatic amylase in the carbohydrate story, or if we looked at trypsin and chymotrypsin in the protein story, or here we're looking at nucleases in the nucleic acid story. So we're getting the idea that the pancreas does produce a large number of digestive enzymes that are really useful to us, but that it actually does its work in the intestines. Because, of course, the pancreas is not actually part of the alimentary canal, but the small intestines are. So we're going to draw the first part of the small intestine, called the duodenum. The duodenum is about the first 10 inches of the small intestine. Just keep in mind that the stomach kind of leads into, through the pyloric valve, it leads into the duodenum. And then we're going to go ahead and draw this little structure on the side of the intestine. This is actually going to be the hepatopancreatic valve, the hepatopancreatic sphincter. And the purpose of this little sphincter is to allow for some digestive juices to enter into the duodenum, the very first part of the small intestine. And so we're going to draw in blue, we're going to draw 2 tubes, one of them coming from the pancreas, and that's going to bring pancreatic juices into the duodenum, such as chymotrypsin and trypsin, such as pancreatic amylase, such as nuclease. And then, in a moment, we're going to be studying another one, which is going to be a substance that's brought in from the liver and the gallbladder. So this hepatopancreatic system allows us to bring things from the liver, the hepatic system, and from the pancreas into the duodenum. And so when these digestive structures come from these 2 places, that is why these digestive structures actually do their work in the small intestine, because they have access through here. The food is then able to remain within the lumen of the alimentary canal, outside of the body, remember, and still have access to these very special structures created inside of these other organs. So let's get back to the nucleic acid story. We're going to draw ourselves a little more clear here, a little more clear nucleotide. We have that central sugar, a pentose sugar in the middle. It would be deoxyribose in this case, but it could be ribose if it was RNA, same thing. A phosphate group off to one side and that nitrogenous base off to the other side. We are going to do our final digestion. We're actually going to take this technically monomer of nucleic acids, and we're going to break it down further because it's still a pretty big molecule and would be quite difficult to absorb. So we have two brush border enzymes. Nucleosidase is going to chop off the nitrogenous base from the sugar, and phosphatase is going to chop off the phosphate group from the sugar, and therefore you have these three separated structures, much easier to absorb at this point. All three are going to be absorbed directly into the bloodstream through the walls of the villus. Right, so our very last of the foodstuffs, we've already discussed carbohydrates, proteins, and nucleic acids. And the most complicated story is that of the lipids. So lipids are nonpolar, and so therefore they kind of tend to cling to each other in a fat globule, and they sort of stick together through the mouth. Immediately they start to find each other because most of the fluids in the body are quite polar, and so these nonpolar structures tend to kind of nonpolar attracts nonpolar, so they tend to kind of stick together, and therefore they're quite difficult to work with. This very massive fat globule needs to be broken down first. So the first thing that's going to happen is we take bile from the liver. It's produced in the liver and it's stored and concentrated inside of the gallbladder, and then we're going to be calling these bile salts or bile detergents. And so technically not an enzyme, but rather some people would actually consider this to be mechanical breakdown rather than enzymatic breakdown, but nonetheless, I think either way works. So we take this very large fat globule and we start to break down smaller pieces of it, and we actually surround the fat globule with bile salts. The bile salts then form a micelle, which is a structure that is polar on the outside and non-polar on the inside, and this allows the fat to exist in smaller fat droplets. The particular name for this actual micelle, micelle is a general term, but this specific micelle is called an emulsion of fats. So fat droplets surrounded by bile salts. Now, this gives access then to the actual enzyme that's going to break down the fat, which is called lipase or lipase. There's actually three places where lipase is secreted. There's some actually secreted in your mouth. There's some secreted by the chief cells in the gastric glands of the stomach. But the main source of lipase in your body is the pancreas. Now take a look at this. This is the first orange that we're using from the pancreas. So this is not a brush border enzyme, although we are drawing it in orange because it is the final digester, if you will. It is the final digester of these fat droplets. So what the lipase does then is it takes these fat droplets, which are small, and breaks it down into its monomer. The monomer is going to be the triglyceride. Triglyceride is the way that most fats are stored, for example, within your blood. Now your body just loves fat. Your body loves fat, and so it's going to be using this fat as for energy. Can break down the fat structures and get a lot of calories out of that. So we go to a lot of trouble to make sure that we're absorbing as much fat as possible. We crave fatty food. We love fatty food. So anyway, these triglycerides then are going to be reformulated again. Triglycerides are mostly nonpolar, so they are able to go straight through the walls of these villus cells here. We are drawing the outside of the villus. Remember that is made of simple columnar epithelium, and so the triglycerides go directly straight through the membrane of these villus cells, these simple columnar epithelial cells, and then they are bonded actually to a protein. This protein is sort of like a chaperone or a carrier, and together, triglyceride and its protein carrier would be called a chylomicron. Now, the chylomicron is going to be transported directly, basically, through the walls of the villus, as we're showing here. It left the lumen as a triglyceride. It was bonded to a protein carrier inside of this simple columnar epithelial cell in the wall of the villus, and is able to make its way directly through the other side and directly into a lacteal, which represents which represents the lymphatic system. And so 3 of the foodstuffs that we're looking at today, carbohydrates, nucleic acids, and proteins, once they're broken down into their monomeric parts, they're able to be absorbed through the wall of the villus and into the blood supply there. But in this case, the fats, because I guess it's the larger structure that's the main reason, the lipids are actually going to be absorbed directly into the lacteal.